mRNA processing. As a gene's DNA is transcribed into primary transcript mRNA, also known as pre-mRNA, you will follow the next step called mRNA processing, in which the same primary transcript mRNA will go through a few processes in order to become a mature mRNA and consequently be able to do its job of synthesis of protein. mRNA processing can be broken down into three processes, named 5' capping, 3' poly A tail, and splicing.
5' capping. The main purpose of the 5' capping is while the transcription is occurring, a 7-methylguanosine cap is added to the 5' end of the rowing transcript by a 5-to-5 triphosphate linkage. Transcription then continues to completion. The cap is a modified guanine nucleotide and it protects the transcript of being broken down. Connected to the mRNA via a usual 5-to-5 triphosphate linkage, the capping enzyme is a complex of three different enzymes: methyltransferase, RNA triphosphatase, and guanylyl transferase that catalyzes the attachment of the 5' cap to the RNA molecules. The cap protects the 5' end of the primary RNA transcript from attack by ribonucleases and is recognized by eukaryotic initiation factors involved in assembling the ribosome on the mature mRNA prior to initiation translation. How does the cap get attached? Firstly, the 5' end of the rowing mRNA strand gets close to the region where the capping complex is sitting. Then, the phosphohydrolase enzyme is in charge of chopping off the gamma phosphate group. Then, the two phosphates are removed from the guanyl and it is added at the 5' end linked by a phosphate group thanks to the guanyl transferase. Finally, the methyl transferase adds a methyl group at the 7th position of the guanine. This will be the final stage of the guanosine cap, or also called the M7G cap, attached to the mRNA. The cap helps with the splicing stability and the translation of the RNA out of the nucleus. It also makes the mRNA be transported to the cytoplasm and be recognized by the ribosome for future attachment to them.
Following the 5' capping, mRNA continues with the 3' poly A tail process. The main idea in this process is to be able to identify the cleavage at the 3' ending and to understand the importance of the polyadenylation. Let's break it into more details. Two specific nucleotide sequences in the mRNA, poly A signal and U or GU rich region, will be binded by cleavage factors. The 3' ending will then move to the correct configuration and stabilizing factors will be added, attaching both of the cleavage factors. After that, an important enzyme called poly A polymerase will attach to the mRNA between the two specific nucleotide sequences, causing the cleavage of the 3' end from the complex. As a consequence, the cleaved piece begins to degrade. At this point, the poly A polymerase immediately starts the polyadenylation step, which is the addition of a string of approximately 200 adenine nucleotides, also known as poly A tail, to the 3' end of the just cleaved pre-mRNA. Additional proteins will also bind to the poly A tail, helping to increase the rate at which the poly A polymerase adds adenine. Once the poly A tail reaches its full length, poly A polymerase is signaled to stop adding adenine and the enzyme is disattached from the complex. The poly A tail is an extremely important structure which provides stability and protection to mRNA against mRNA degradation by cellular enzymes. Due to this stability and protection, poly A tail allows further movement of the mRNA from nucleus to cytoplasm through the nucleopore and further attachment to ribosomes. The 3' poly A tail process is completed and now ready to undergo splicing.
In
RNA splicing, specific parts of pre-mRNA called introns are recognized and removed by a protein and RNA complex called the spliceosome. Introns can be viewed as junk sequences that must be cut out so the good parts of the RNA molecule can be assembled. But what are the good parts? The pieces of the RNA that are not chopped out are called exons. The exons are pasted together by the spliceosome to make the final mature mRNA that is shipped out of the nucleus. Introns are removed from primary transcripts by cleavage at conserved sequences called splice sites. These sites are found at the 5' and 3' ends of introns. Most commonly, the RNA sequence that is removed begins with the dinucleotide GU at its 5', known as the 5' splice site, and ends with AG at its 3' end, known as the 3' splice site. These sequences are known to be critical because changing one of the nucleotides results in inhibition of splicing. Another important sequence occurs at what is known as branch site A, located anywhere from 18 to 40 nucleotides upstream from the 3' end of an intron. The branch point always contains an adenine, but the rest of the code does not really matter as long as it starts with adenine. Spliceosomes recognize the sequences at the splice sites. Splicing occurs in several steps and is catalyzed by small nuclear ribonucleoproteins, snRNPs, commonly pronounced SNRPs. Spliceosomes are made up of these SNRPs, and all the SNRPs are U1, U2, U4, U5, and U6. First, the pre-mRNA is cleaved at the 5' end of the intron following the attachment of a SNRP called U1 to its complementary sequence of GU within the intron and U2 to A or branch site. Then U5, U4, and U6 join the bound U1 and U2, and the spliceosome is assembled, giving it a almost loop shape. Then U1 and U4 are expelled, and U6 is paired with the 5' splice site and U2. The 5' intron is then cleaved. The cut end then attaches to the conserved branch point region downstream through pairing of guanine and adenine nucleotides from the 5' end and the branch point A, respectively, to form a loop structure known as a lariat. The bonding of a guanine and adenine base takes place via a chemical reaction known as transesterification, in which a hydroxyl OH group on a carbon atom of the adenine attacks the bond of the guanine nucleotide at the splice site. The guanine residue is thus cleaved from the RNA strand and forms a new bond with the adenine. Next, the SNRPs U2 and U5, U6 appear to contribute to positioning of the 5' end and the branch point in proximity. With the participation of U5, the 3' end of the intron is brought into proximity, cut, and joined to the 5' end. This step occurs by transesterification. In this case, an OH group at the 3' end of the exon attacks the phosphodiester bond at the 3' splice site. The adjoining exons are covalently bound, and thus the resulting lariat is released with U2, U5, and U6 bound to it, and you now have a mature mRNA. There is also something known as alternative splicing. Alternative splicing is a splicing process resulting in a varying composition of exons in the same RNA and creating a range of unique proteins. Alternative splicing of pre-mRNA is an essential mechanism to enhance the complexity of gene expression, and it also plays a vital role in cellular differentiation and organism development. Alternative splicing enables exons to be arranged in a different combination where different configurations result in different proteins. The process of alternative splicing might occur either by skipping or extending some exons or by retaining particular introns, resulting in different varieties of mRNA formed. Splicing enhancers. These are splicing signals that promote splicing and lie within exons and are known as exonic splicing enhancers, also known as ESEs. ESEs are bound and activated by splicing factors known as SR proteins that are thought to act by recruiting core splicing factors such as the U1 SNR SNRPs or U2AF to appropriate splice sites. There are also things known as splicing repressors. Splicing repressors or silencers are sites to which splicing repressors protein bind, reducing the probability that a nearby site will be used as a splice junction. These can be located in the intron itself or in a neighboring exon. They vary in sequence as well as the type of protein that bind to them.
Now we are gonna do a small comparison to bacteria. First, while the eukaryotic mRNA is monosystronic, it only has one site for initiation and termination of protein synthesis. On the other hand, the bacterial mRNA is polysystronic, where a single mRNA strand is transcribed by several structural genes. In bacteria, the translation of the mRNA begins while the DNA is still transcribed from the DNA molecule, while in eukaryotes, it is first transported to the cytoplasm via the nuclear pores, then is attached to the ribosome, and finally, the protein is synthesized. Also, the eukaryotic mRNA have much longer lifespan compared to the lifespan of the prokaryotic mRNA. This one is very short. And finally, the eukaryotic mRNA undergoes major post-transcriptional changes like the capping, poly-A-tail, and splicing, the processes that we just talked about, while in bacteria, the post-transcriptional changes are very little.